Higher Level Biology for the IB Diploma Program, Pearson book, 3rd edition, Theme C. Now we're into Chapter 4.1. So Theme C was Interaction and Interdependence. Chapter 4 is Ecosystems. All right. C4.1 Populations and Communities. The guiding questions. How do interactions between organisms regulate sizes of populations in a community? What interactions within the community make its populations interdependent? In this chapter, we will see that nature has multiple ways of ensuring that no single species takes over an ecosystem, and that each population has the potential to reach its maximum, but that many factors can limit populations or decrease their numbers. Such factors include the availability of food and water, presence of predators, and the introduction of diseases. Some organisms engage in chemical competition by secreting molecules into their environment to reduce the chances of their competitors being successful. No species lives in an isolated bubble. Plant eaters need plant material to survive, carnivores need other animals to eat, and some plants rely on microorganisms to provide nutrients such as nitrogen compounds to help them grow. This last example shows that sometimes two very different species can cooperate for mutual benefit. C4.1.1 Populations. Populations as interacting groups of organisms of the same species living in an area. Students should understand that members of a population normally breed, that means mate and reproduce, and that reproduction isolation is used to distinguish one population of a species from another. In biology, a population is defined as a group of individuals of the same species living in the same geographical area at the same time and able to interbreed. Here are a few examples of populations. Emperor penguins in Amundsen Bay, Antarctica, bush crickets living in a grassland in southern Europe, rainbow trout along the northwest coast of the United States, dandelions growing in the same grassland as the bush crickets. Okay, so see how it was a specific group of individuals of a species living in a specific area. Consider the emperor penguins. There is more than one population of them in Antarctica. How do we know where to draw the line between one population and the next? We can ask how probable it is that the individuals in population A might interbreed with individuals in population B. If penguin population A is separated by hundreds of kilometers of ice and open sea from population B, it is much more likely that penguins will stay and interbreed in their own population. We would be less sure of this if the populations were geographically closer to each other or if penguins could fly. When studying ecology, we can measure many things about a population. The most obvious is population size, which is the number of individuals. But populations are not fixed. They are dynamic. The size changes. So we can also measure the change in population over time, which could go up or down as a result of factors such as immigration and death. We will see that estimating the size of a population is not an easy task. Other characteristics that can be used to describe a population are population density, its geographical distribution, and the maximum number of individuals that can be supported by the resources available. Once we understand a population, we can start to look at how it interacts with its environment and other species. C4.1.2 Estimating population size. Estimation of population size by random sampling. Students should understand reasons for estimating population size rather than counting every individual, and the need for randomness in sampling procedures. Nature of science. Students should be aware that random sampling, instead of measuring an entire population, inevitably results in sampling error. In this case, the difference between the estimate of population size and the true size of the whole population is the sampling error. The estimate is 46 and the true size of the whole population is 50, then your sampling error is 4. Ideally, to know the size of the population we are studying, we would count every last individual. For emperor penguins, this would involve going to a Hudson Bay in Antarctica and counting each one. What would that look like? Once you started counting, how could you be sure that you had not already counted a particular penguin? Some members of the population will be chicks, keeping warm near their parents, and it will be hard to see all of them. Some individuals may be out in the water getting food. These are just a few examples of how most of the time it is simply impossible to count every last individual in a population. Instead, we need to rely on estimates. We count a sample and use that sample to estimate the overall population size. There are 2 types of sampling: systematic and random. Systematic sampling is when a line or a grid is set up and the measurements or counting are carried out only at specified regular intervals. For example, a 50-meter measuring tape can be laid across a rocky shore, and seaweed or snails can be counted in a 1 square meter area every 5 meters. Random sampling is when randomly chosen zones of the population's geographic distribution are sampled. Random directions and random distances between samples are used to try to overcome any bias that the investigators might have that would favor a particular area. Random sampling using quadrats and a method of mark and recapture are discussed in sections C4.1.3 and C4.1.4. Here's a picture of a biology student using a quadrat on a rocky shore. Random sampling inevitably results in sampling error. The difference between an estimated population size and the true size of the whole population is the sampling error. If ecologists randomly take 200 samples from a prairie that are 1 square meter each and determine that the average number of ferns in each square meter is 1.3, they can deduce that if the prairie is 10,000 square meters, the total population should be 13,000 ferns. But this assumes that the ferns are evenly distributed across the prairie. If there were actually only 8,000 ferns in the area study, the sampling error would be quite large. 5,000 extra ferns were incorrectly estimated. This is one of the many reasons why, when given a number, scientists often ask how it was determined and what the degree of precision is. They want to know how much error might be involved and how close the estimate is to the true value. Here is an allegory to illustrate sampling error. A man gets in a boat and rows out into the ocean. He fills the bucket with seawater and looks in the bucket to count the number of fish he sees. He sees none and declares, "There are no fish in this ocean." C4.1.3 Random quadrat sampling to estimate population size for sessile organisms. Both sessile animals and plants, where the numbers of individuals can be counted, are suitable. Application of skills. Students should understand what is indicated by the standard deviation of a mean. Students do not need to memorize the formula used to calculate this. In this example, the standard deviation of the mean number of individuals per quadrat could be determined using a calculator to give a measure of the variation and how evenly the population is spread. Standard deviation is a quantity that measures the average distance between the measured values and the mean of those values. It describes how spread out the data are in relation to the mean. Random sampling can be used to estimate population size for organisms that stay in one place. For example, plants, lichens, and corals. Such organisms are referred to as sessile. For much of their lives, they do not change location. The example in the ebook shows how to estimate the population size of a sessile organism using a quadrat. A quadrat is a square of a particular dimension that can be made of a rigid material such as metal, plastic, or wood. Using a quadrat means the surface area of the sample size is the same for each count you take. C4.1.4 Sampling motile organisms. Capture, mark, release, recapture, and the Lincoln Index to estimate population size for motile organisms. Application of skills. Students should use the Lincoln Index to estimate population size. Population size estimate equals M times N divided by R, where M is the number of individuals caught and marked initially. N is the total number of individuals recaptured, and R is the number of marked individuals recaptured. Students should understand the assumptions made when using this method. The capture, mark, release, recapture method, all of that is bolded, you need to know this method, is a sampling technique that enables you to estimate the number of animals in an ecosystem. It is used instead of quadrats for motile organisms, those that move around. The technique involves catching some of a population and marking them. The marked animals are released back into the ecosystem and given a suitable period of time to remix with others in their population. A second sample of the population is then captured. Some in the second sample will be marked and some will be unmarked. The proportion of marked to unmarked individuals in the second sample is assumed to be the same as the proportion of the originally marked individuals to the whole population. The Lincoln Index is then used to estimate the number of individuals in the population. Here is the formula. The total population is equal to the number of individuals caught and initially marked multiplied by the number of all individuals recaptured divided by the number of marked individuals recaptured. The Lincoln Index can be written using variables instead of words. Total population equals M times N over R, where M is the number of individuals originally caught and marked, N is the total number of individuals recaptured, and R is the number of marked individuals recaptured. The capture, mark, release, recapture method has some limitations. You need to know these limitations. Capturing and marking the animals may injure them. The mark may make an animal more visible to predators, and if the marked animals are eaten, the second sample will not be reliable. It assumes that the population is closed with no immigration or emigration. The last point is a rather big assumption, because very few populations are closed. Other assumptions are that all the individuals in a given area have an equal chance of being captured, that marked individuals will be randomly distributed after release, and that marking individuals will not make them more or less likely to be recaptured. The mark and recapture technique can be used by experts to estimate the size of a bird population. Small metal bands with identification codes can be attached to a bird's leg to tag it, And when the same bird is recaptured, population ecologists know where and when it was originally captured. C4.1.5 Carrying Capacity. Carrying capacity in competition for limited resources. A simple definition of carrying capacity is sufficient, with some examples of resources that may limit carrying capacity. No habitat can accommodate an unlimited number of organisms. Populations often grow, but they cannot continue to grow forever. There comes a time in the growth of a population when its numbers stabilize. This number, the maximum number of individuals that a particular habitat can support, is called the carrying capacity, and it is represented by the letter K. Consider, for example, a given zone in a forest. There is a maximum number of trees that can grow there. This number is attained when enough trees are present to catch all the sunlight, leaving every square meter of the forest floor in shade. New tree seedlings trying to grow under the adult trees will have difficulty getting any sunlight. In tropical rainforests, such as this one in Costa Rica, very little sunlight reaches the forest floor because a maximum number of trees and plants have spread their leaves to catch all the sunlight they can. Young trees can store energy for years with very little vertical growth, waiting for a larger tree to die, leaving a hole in the canopy. The young trees then compete to use the opening to take the old tree's place. Those that lose this competition usually die. For a young tree to join the mature population, an old tree must die. Hence, there is no net increase in the tree population, at least at the canopy level. A limiting factor is something that can prevent a population from getting bigger or reduce a population's size. So in this example, it's access to sunlight is the limiting factor. Limiting factors that define the carrying capacity of a habitat include the availability of resources such as water, food, sunlight, shelter, space, and oxygen. The latter notably in aquatic habitats. So oxygen is limiting in the water. The buildup of waste, such as excrement and excess carbon dioxide, predation, and disease. C4.1.6 Negative feedback. Negative feedback control of population size by density-dependent factors. Numbers of individuals in a population may fluctuate, going up and down, due to density-independent factors, but density-dependent factors tend to push the population back towards the carrying capacity. Okay, so a population can change due to things that have nothing to do with the population density. However, factors that do depend on density are usually limiting. In addition to the competition for limited resources, include the increased risk of predation and the transfer of pathogens or pests in dense populations. As a population grows, its population density increases. How many individuals there are per square meter. There will be more individuals per unit of habitat, such as more plants per square meter of prairie or more plankton per liter of seawater. Some factors that change the size of a population are dependent on the density of the population, such as the spread of disease. These are called density-dependent factors. They tend to keep a densely populated area at or below its carrying capacity. Other factors are density-independent, meaning that it does not matter if there is a sparse population or an overcrowded population. Examples of density-independent factors include climate change, or a nearby forest fire, or a volcanic eruption, maybe an earthquake. Populations both big and small will be affected, and the change in numbers can be an increase or a decrease depending on whether the factor helps or hinders a population. Complex systems require feedback mechanisms to keep parameters from reaching dangerously high or low levels. Positive feedback in a population is when something increases the population, and the system encourages more of the same. For example, in a breeding population that is initially small, the more individuals that are produced by successful breeding, the more individuals there are to produce the next generation. The higher the population density, the higher the chances of a male encountering a female to produce even more individuals in the population. Positive feedback will help a population grow, but it can get out of control. Negative feedback prevents a system from going too far in one direction. If a population continued to grow uncontrollably, for example, individuals would start to run out of resources such as space, food, or water. The chances of diseases spreading also increase when population sizes are large and density is high. The chances of attracting predators to an area increases as the number of prey individuals increases. Negative feedback in the form of competition for resources and the spread of disease works to control the size of the population so that it cannot go above its carrying capacity. When it comes to population dynamics, positive feedback can be a dangerous thing, leading to food shortages and the spread of disease, whereas negative feedback helps prevent such density-dependent issues from getting worse and threatening a population. In a positive feedback loop in populations, more offspring grow up and produce additional offspring, which in turn produce even more offspring. In a negative feedback loop, something such as a paucity of food or the introduction of a disease will regulate the population and slow down or reverse population growth. C4.1.7 Population Growth. Population growth curves. Students should study at least one case study in an ecosystem. Students should understand reasons for exponential growth in the initial phases. A lag phase is not expected as a part of sigmoid population growth. Nature of science. The curve represents an idealized graphical model. Students should recognize that models are often simplifications of complex systems. Application of skills. Students should test the growth of a population against the model of exponential growth using a graph with a logarithmic scale for size of population on the vertical axis and a non-logarithmic scale for time on the horizontal axis. In 1980, the ecosystems around Mount St. Helens were destroyed when the volcano erupted in a sudden, violent blast. The devastation is shown in the picture on the left. Contrast this with the picture on the right, where a few of the dead tree trunks can still be seen, as well as smaller trees that have grown since the eruption. In 1980, there was a major volcanic eruption of Mount St. Helens on the west coast of the United States. After the massive event, little was left of the forest and rivers that had existed on and around the mountain. Forest fires and hot gases burned everything in sight. Volcanic ash rained down, smothering the destroyed forest and covering the carcasses of the animals that had died there. Many species that could escape fled the area. Yet, within months of the eradication of an ecosystem, life was back. Seeds dropped by birds or blown in by the wind germinated in the fertile volcanic ash. Little by little, insects, then birds, then small mammals moved in. Within a couple of decades, a grassland and shrub ecosystem had reappeared. Today, thousands of species flourish in what had been a desolate landscape. If we look at the tree species now present on Mount St. Helens, such as conifers and the red alder, their populations have increased over the decades, but some experienced a decrease in growth when the North American elk populations started to thrive there too. Attracted by new food sources, elk browsed, meaning they ate, the young tree saplings of deciduous trees and conifers, slowing the growth of the tree populations. However, the elk droppings, their feces, also helped improve the soil quality, so other vegetation could grow better. These complex interactions are part of the reason why an ideal population growth curve, like the one shown in Figure 1, almost never exists in nature. This is an idealized population curve showing growth over time following a sigmoid shape. Phase 1 represents the exponential growth phase, Phase 2 the transition phase, and Phase 3 the plateau phase or stationary phase in which the population reaches its carrying capacity. The sigmoid, which means S-shaped, curve of the graph in Figure 1 shows the 3 stages of population growth. The exponential phase, also called the logarithmic phase, during which the number of individuals increases at a faster and faster rate. The transitional phase, during which the growth rate slows down considerably, the population is still increasing, but at a slower and slower rate. The plateau phase, or stationary phase, during which the number of individuals stabilizes and there is no more growth. So what causes the 3 different phases of a population growth curve? The exponential phase. In ideal conditions, a population can double in size on a regular basis. Not counting mortality, for example, a population of bacteria can theoretically double its population every few hours. 1, 2, 4, 8, 16, 32, 64, 128, so on. Without predators, introduced species such as cane toads in Australia can take over habitats with uncontrolled population growth. The reasons for this first phase of exponential growth are plenty of resources such as food, space, and for photosynthetic organisms, light, little or no competition from other inhabitants, favorable abiotic factors such as temperature, and for aquatic organisms, dissolved oxygen levels, little or no predation or disease. The traditional phase. Eventually, after the exponential increase in numbers of individuals, some of the factors listed above no longer hold true. This leads to the transitional phase. The causes of the transitional phase are, with so many individuals in the population, there is increasing competition for resources. Predators, attracted by a growing food supply, start to move into the area. Because of the large number of individuals living together in a limited space, opportunities for diseases to spread within the population increase. The plateau phase. Consider the land around a volcano such as Mount St. Helens slowly being taken over by vegetation within months of a deadly eruption that wiped out all nearby life. Once all the fertile volcanic ground is covered with plants, the space available will be occupied to its maximum. Thus, there is gradually less and less and less available space for any seeds produced by the plants to germinate, and the number of plants stabilizes. With increasing numbers of herbivores, there is a limited supply of food. In response to limited food supplies, animals tend to have smaller numbers of offspring. Some may leave the area and immigrate to a place where there is more abundant food. Predators and disease increase mortality, and the growth curve tends to level off. In this phase, the number of births plus the number of immigrations is balanced by the number of deaths plus the number of emigrations. It's moving out. Nature of science. The population growth curve represents an idealized graphical model. Models are simplifications of complex systems. They help us conceptualize phenomena that otherwise are difficult to understand. They are useful in helping our brains grasp difficult concepts, but because they are simplifications, they have their limits. Modeling population growth. Modeling of the sigmoid population growth curve. Application of skills. Students should collect data regarding population growth. Yeast and duckweed are recommended, but other organisms that proliferate under experimental conditions could be used. C4.1.9 Communities. A community has all of the interacting organisms in an ecosystem. Communities comprise all the populations in an area, including plants, animals, fungi, and bacteria. A community is a group of populations living and interacting with each other in an area. Examples include the soil community in a forest and the fish community in a river. In ecology, the term interacting can mean one population feeding on another or being eaten by another. It can mean that one species provides vital substances for another, as in the case of certain bacteria that can help plants obtain nitrogen from the air. It can also mean that one species is protected by another, as in the case of aphids protected by ants from predator attacks. Interacting can also mean that one species relies on another for its habitat, as is the case for parasites living on or inside the bodies of other animals.